Hello, dear NEET Cognition Team doctors. Today we are going to see NEET Neural Log Series Day 5. This is a live for 30 minutes where you can join with me, write and say it out and lock in your memory. So yesterday was Day 4. We saw Block 1 endomembrane system, the organelles of the endomembrane system, that is endoplasmic reticulum, Golgi bodies, lysosomes and vacuoles. Next, under lysosomes, one thing we missed is the mitochondria, peroxisomes and chloroplast are not included in this because their functions are not coordinated with the endomembrane system. Next is lysosomes. We what do we study? We studied yesterday acidic pH, hydrolytic enzymes, then about Golgi apparatus. It is formed from Golgi apparatus. Next is block 3, nucleus discovered by Robert Brown in 1831. If you have watched yesterday's class, check yourself whether you are able to remember the points. See, I am not seeing the cheat sheet which I have made. I am just reading it out, which means it has gone inside my memory. Okay. So check yourself whether you are able to say all the points which we did yesterday. The nucleolus, it is present in the center of the nucleus. What is the function of nucleolus? RNA synthesis, that is ribosomal RNA synthesis. Then we have nucleoplasm, which is made up of chromatin, that is chromatin and nucleolus. Nucleoplasm is also called as nuclear matrix. Today we are going to see a very interesting topic. It's Day 5 live. Before that, I am going to ask you one question. See, are you able to store water? Can humans store water? I want an answer for this question. If you know the answer, type it in the comment section. But how do plants store water? How do plants store water? There is a structure inside the plant which helps the plant to store the water. Can you name the structure? There is one clue. It occupies the 90% of the plant cell. See, inside the cell you have lot of organelles, right? Mitochondria is very important, chloroplast very important, organelle, ribosomes, nucleus, the brain of the cell. But this organelle, which is a storage organelle, occupies 90% of the plant's cell. What is the name of the structure? If you know the answer, quickly type it in the comment section. Next is the block 1. The answer is vacuole. Okay. In block 1 we are going to see about vacuole, and in block 2 we are going to study about semi-autonomous organelles. What is the meaning of semi-autonomous organelles? Can you name the organelles? There are similarities between those two organelles. Do you know those similarities? So today we are going to see about semi-autonomous organelles. They are mitochondria and chloroplast. And the similarities between mitochondria and chloroplast. See, they are totally two different organelles. What is the function of mitochondria? Very easy. ATP synthesis. They are the powerhouse of the cells. Then chloroplast, synthesis in photosynthesis. But they do have few characteristics in common. Okay, quickly, can you tell me what 4 characteristics? If you know the answer, even one, you can type it in the comment section. So shall we start, students? First is the vacuole. In block 1, we are going to see about vacuole. See, you have plant cell. Okay. Just a small diagram for you to understand plant cell. Plant cell is what type of cell? An eukaryotic cell. Inside the plant cell you have other organelles which are very important for the plant cell. Then you have a larger structure which occupies 90% of the plant cell. You call that structure as vacuole. What are the contents of vacuole? That itself is an MCQ. Contents of vacuole is an important MCQ. We will cover in another blog. Okay, today the basic important points about vacuole we are going to do. Then this is the cytoplasm. Okay, you have other organelles. So there is a separation between the cytoplasm, that is the matrix, cell matrix, and the vacuole by means of a single membrane. This membrane is only one layer, single membrane. This is very important need. What is the single layer of membrane which surrounds the vacuole? You call it as tonoplast. Call it as tonoplast. Okay, this is the first point. Then second point is this vacuole forms the 90% of the cell volume. We saw it now. Then what are the two types of vacuoles which is mentioned in NCERT? One is the food vacuole, which is seen in protista. These are examples. This is important for your match the following. Then you have contractile vacuole, which is seen in amoeba. Okay, so what is the function of this contractile vacuole? It helps in osmoregulation and excretion in amoeba. What is the meaning of osmoregulation? Meaning of osmoregulation is water balance. Okay, so inside the amoeba cell there should be homeostasis. What is the meaning of the word homeostasis? If you know, type it in the comment section. Homeostasis means maintaining the balanced state of the cell. If there is some disruption, then the cell will die. Okay, so if there should not be too much of water nor less. This should be maintained. Okay, because amoeba is seen in water, fresh water, the first lesson. Okay, so there will be too much of water entering into amoeba. Okay, so this contractile vacuole will detect that and it will expel the water, excess water, into the surrounding. Okay, so this is how contractile vacuole helps in osmoregulation, and it also helps in excretion. Then this is food vacuole. See, there is a difference between food vacuole and contractile vacuole. Just for interest, you can know, it is not mentioned in NCERT. They have given only the examples. Food vacuole is, the name itself says food vacuole. So it must be to engulf the prey, engulf the foods, bacteria or other organisms. Okay, here it is food. This is food means it approaches and it surrounds and it takes inside the cell. So this is like engulfing. Okay, engulfing. This is called as the food vacuole. These are called as temporary stomachs. Temporary stomachs. Okay, this you need not remember. Just for an interest, I will share this to you. Now let us quickly see this. I hope now you will understand these points. The single layer membrane of vacuole is called as tonoplast. Plant cell volume has vacuole which occupies 90% of the cell volume. Next is the example. Food vacuole is seen in protista and contractile vacuole is seen in amoeba. Contractile vacuole helps in osmoregulation and excretion. Now we are going to write it 3 times. After finishing this, you have to write it 2 more times without seeing the original cheat sheet. Okay, shall we follow each other? Okay, see, when you are writing, practicing, highlight this ST, which means tonoplast made up of single membrane. This is an important MCQ. Tonoplast, the single membrane surrounding the vacuole is called as tonoplast. The single membrane which surrounds the vacuole is called as the tonoplast. The single membrane which surrounds the vacuole is called as the tonoplast. I'll practice writing like this itself. Okay, but don't write in your theory exam like this. Just for you to remember for a while. After that, you will automatically remember. Next is the plant vacuole, which occupies 90% of the cell volume. How much? This is a numerical value. 90% of the cell volume. How much percentage? 90% of the plant cell volume is occupied by the vacuole. 90% of the cell volume is occupied by the vacuole in a plant cell. This is very important. This much of volume is seen only in plant cell. Okay? Because it stores water. Next, food vacuole is seen in protista, which helps the protista to engulf the organisms. That is the food. Food vacuole is seen in protista. Third time, food vacuole is seen in which organism? Protista. Did you complete, students? Next is the contractile vacuole, seen in amoeba. Contractile vacuole, contractile, you can write this word alone. Vacuole you don't need to write. Okay, seen in amoeba. Contractile vacuole is seen in amoeba, because vacuole is a common word, you know, but contractile is bit different. Okay, so write this part alone and you can save time for this. What are the uses of contractile vacuole? Osmoregulation. What is the meaning of osmoregulation? Just now I shared you osmoregulation. Quickly type the answer in the comment section. Osmoregulation means water balance. Water and electrolytic ions should be kept balanced in a balanced condition inside the cell. Osmoregulation. Next, another function is excretion. Contractile vacuole helps in excretion of amoeba. Excretion in amoeba. Excretion in amoeba. Contractile vacuole helps in excretion in amoeba. Okay. Next we see the block 2, that is semi-autonomous organelles. Okay. In what are the two semi-autonomous organelles? That is mitochondria and chloroplast. Meaning of semi-autonomous. Semi-autonomous. What is the meaning of autonomous? Means acting on its own. Yeah, now I have a physiology question for you. Which organ shows autorhythmicity? Type the answer in the comment section. Which is the organ which shows autorhythmicity? No? Which beats continuously when you sleep or when you are awake, when you do some work. Throughout the day it keeps beating. What is the organ? So leave the answer in the comment section. Which means autorhythmicity. Here it is acting same way, it is acting on its own but not completely. It is acting only partially. Okay, so this is called as semi-organelles of the cell or mitochondria and chloroplast. Very important. This question is asked in assertion reason type, statement type, or it can be asked in match the following. Mostly this is AR type of question. Okay, why they are called a semi-autonomous mitochondria and chloroplasts are called a semi-autonomous organelles. This can be assertion. The reason why they are called a semi-autonomous organelles, because they have their own circular DNA and they have their own 70S ribosomes. This two, if you see these two words in the reason, then you can easily choose the answer. Okay. Now what are the we'll write it 3 times. Semi-autonomous organelles or mitochondria and chloroplast. Semi-autonomous organelles are the two organelles are mitochondria and chloroplast. These semi-autonomous organelles are mitochondria and chloroplast. So now these, I already told you, these two are two different organelles with two different function, but how they are connected. Okay, they have four characteristics in common. The first is both are double membrane bound organelles. How many membranes? Two membranes. Double means two layers. Okay, the rod shaped mitochondria, outer membrane, inner membrane. This inner membrane folds to form what? Cristae. Okay, now I have a question for you. The inner membrane's folding is called as cristae. What is cisternae? What is cisternae? The question is, what is cisternae? Find out the answer, or if you know the answer, quickly type it in the comment section. Okay, so mitochondria and chloroplast are double membrane bound organelles. They are two layered organelles. Double membrane bound organelles are mitochondria and chloroplast. Now we saw the reason why they are called as semi-autonomous organelles. They have their own circular DNA and 70S ribosomes. Only because of this reason they can take care of themselves. So what is the type of DNA present inside mitochondria and chloroplast? It is circular type of DNA. Now the question is, what is the shape of nuclear DNA? Quickly type the answer. Shape of the nuclear DNA. I told you, no, DNA has, nucleus has got DNA. What is the shape of nuclear DNA? Circular DNA is seen in mitochondria and chloroplast. What is the shape of nuclear DNA? Now again, getting back to this, circular DNA is seen in mitochondria and chloroplast. Both organelles have circular DNA. Circular DNA. Okay. Next, we saw that they have ribosomes. What type of ribosomes? 70S. What does S mean? Already we studied this. Swedberg's unit. We finished this, right? If you are able to remember, I'll be very happy. You have scored marks for that. Then 70S ribosomes. What type of ribosomes are seen in mitochondria and chloroplast? 70S types of ribosomes. Okay. Now, prokaryotic organism has what type of ribosome? Already we saw this in the previous days. 70S. 50S bigger unit and 30S smaller unit. Totally 70S. Okay, we saw that we shouldn't add because it is a sedimentation coefficient. So prokaryotic, or this is very important, prokaryotic organism also has 70S ribosome, and the ribosomes inside the mitochondria and chloroplast also has 70S ribosomes, which means there is some link between these two, isn't it? See, you have a prokaryote. This is prokaryote. Okay, prokaryotic cell, a bacteria or whatever. One prokaryotic cell. This is a eukaryotic cell. Eukaryotic cell. Okay, inside this eukaryotic cell you have mitochondria and chloroplast. This what is the ribosome here? 70S type of ribosomes. Ribosomes for mitochondria and chloroplast, just now we saw it contains 70S, 70S. Isn't it? But what does the cytoplasm ribosome? A eukaryotic cell will have a ribosome, right? That ribosome, what type of ribosome is this? This is 80S type of ribosome. This is very important NEET trap question. I'll explain you later. Now coming to this point, okay, this prokaryotic cell also has 70S, mitochondria and chloroplast inside an eukaryotic cell also has 70S, which means they say once it was a prokaryotic cell. These two was once a prokaryotic cell which came inside the eukaryotic cell as an organelle. Okay, this is what is called as endosymbiotic theory. Just remember the name alone. Okay, because I have to explain the ribosomes. I explained this also, endosymbiotic theory. Just remember the name alone, okay? So what are the two organelles which shows relationship to endosymbiotic origin? Mitochondria and chloroplast. Shall we write? Endosymbiotic origin. You should write this word many times. Then you can remember endosymbiotic origin. Endosymbiotic origin. Okay. Now, shall we quickly see the similarities? Both mitochondria and chloroplast have double membrane, that is 2 layers, circular DNA, 70S ribosomes, and endosymbiotic origin. All these 4 are shared by both mitochondria and chloroplast. The 70S ribosomes are they show similarity with the prokaryotic ribosomes because both are of 70S type. Okay. Now the question, I explained you already. See, now I am going to ask you the trap question. Okay, NEET Examiner Trap. It is a ribosome location trap. I already explained you. Now if you are able to find out the answer, it means I have explained it clearly and you have focused it with full concentration. See, NEET Examiner Trap is ribosome location. That is, suppose if the sentence says eukaryotic cells contain only 80S ribosomes. Eukaryotic cells contain only what type of ribosomes? 80S ribosomes. What will you choose? Is it yes or no? It is no. That is, the statement is false. Because what is the fact here? See, inside the eukaryotic cell, the cytoplasm, there is a ribosome, that's a separate organelle, you can see in the eukaryotic cell, right? So that cytoplasm is 80S type. The ribosome of mitochondria is 70S type. Chloroplast is 70S type. So inside a eukaryotic cell, the ribosome organelle, I'll write it down, ribosome organelle is 80S. Mitochondria and chloroplast are 70S, which means eukaryotic cells contain both 80S and 70S type of chromosomes. Okay. Now, I hope you got the point. Okay. Now, moving to primary function of mitochondria and chloroplast. So, before going into that, we have crossed 24 minutes. I want to share you one thing. Which I feel today, okay, that is, let me get a fresh piece of paper. So how was the day? Did you all had your snacks to fuel your brain, which is very important before the evening session? Particularly proper hydration is important. Did you all hydrate yourself? This is not straightly related to the topic, but it is related to our studies. That is, it's a mental hack. Okay, it's not a tip, it's a hack. That is, I'll take myself, okay, whatever I feel I'm sharing to you, that's all. Day 1 to 5. Okay. Actually, I am very good at communication, but still I have some sort of hindrance, okay, in doing live because of my health issues, that is withdrawal, okay. We'll leave that, but still my mind has got confused with that, okay. That is a temporary situation. I can retrain my brain. This is very important. What do you know? There is something called as neuroplasticity. Now tell me the cells of the brain. They are called as neurons. Okay, the structural and functional unit of the nervous system. Neurons, they have hand-like structure, you call as dendrites, a long axon, then terminals. then again one neuron connection with the dendrite, then like this it goes on, because impulse transmission happens, it jumps and it relays. So all the neurons, for example, like this, throughout the brain you have neurons, right? All are interconnected like this. So if you have an old habit, for example, if you have an old habit, you can definitely reframe it to a new habit. That is rerouting, changing the route, the old habit route into new habit. This happens when you do it repeatedly, which means repetition. You call it as repetition. For example, if you say you have difficulty in doing physics problem, first change your mindset. You say hard, fearful thing, your brain will also go fixes a mindset that it is going to be hard. So why does fear comes? It's because lack of confidence. Why do you have lack of confidence? Because you have not prepared enough, isn't it? What is preparation? Understanding and repeating. What we are doing in recall, right? Repeating, you repeat yourself, so that you will never ever forget it. Okay, the best way is to go teach someone. See, when I am teaching you, it's like I am learning even more, even about endosymbiosis. When I studied about endosymbiotic endomembrane system. Yesterday when I studied about endomembrane system, automatically I can understand how an insulin secretion happens. Okay, it's because of endomembrane system only. Endomembrane system. So when you read 50 times equal to writing 10 times equal to teaching 1 time. If you teach once, it is equal to reading 50 times. You're all reading, right? As you're reading, keep on reading. But have you ever draw a biology an eukaryotic or prokaryotic cell? No, because you feel it is time consuming. So you just look at your eyes and you read the names and you go, because diagrams are very, very important. I'll make a class of how to study diagrams. With the diagrams alone, you can finish off writing the whole theory. That's how I did in my physiology first year. Okay, I'll tell you an example. See, these are the kidneys. Okay, then you have ureters, urinary bladder, urethra passage. The reflex, you call it as micturition. Here it is for the storage. This is for the collecting tubes, draining tubes. It collects the urine, and here, and right and left side kidneys, both two kidneys, they are paired. You have to remember only the numerical values related to this. One is placed a bit lower than the other. The reason for that. Okay, if inside you have adrenal cortex, adrenal medulla, only two hormones. See, with the figure, I am able to elicit all the points. Okay, so you have to see diagrams are very important in biology. Now, the point which I want to mention, stress here is repetition. Repeat. Because today I thought, why I feel that hindrance? Why I have that hindrance? Because I feel like I am going for a live. What if I say something wrong due to my withdrawal? What if I am not able to memorize everything when I am doing live? All these things, okay, acts as a hindrance in my subconscious mind. Okay, so if I think like I am not going to bother about anyone, I am going to do for my progress. I am going to learn more. I am going to equip myself. How that comes is by repetition. I have to speak some 10 times as practice before I take the class. Isn't it? So, whatever, if you forget physics, formulae, chemistry, equations, write it as many times as possible, and I'll challenge you, will never ever forget, provided you should understand the concepts well. You should not write something like x plus y means they'll write like x, then plus 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 y. Now it should not be like this. What is the value behind this, and what will come here? You should understand the concepts, and then you have to practice with the repetition, because every master was once a beginner. How he has become master? Because he repeated it as many times as possible, which many people fail to do. If you go and ask the toppers, the secret will be this. One of the secret, I'll say, there is not one way for success. You have lot of ingredients to make a good recipe. So success is a one which you have to add many ingredients to make it as a flavorful recipe. One is repetition. Okay, repetition and recall. Okay, so we'll get back to the class. I just want to share this with you. Now, what is the primary function of mitochondria? Aerobic respiration. What is the function of mitochondria? It is aerobic respiration. What is the function of chloroplast? Photosynthesis. Photosynthesis. Now, photosynthesis means sugar synthesis. Okay. Aerobic respiration means conversion of sugar to ATP, that is energy. This is synthesis of sugar. This is breakdown of sugar. Understood? Photosynthesis is synthesis of sugar. This is breakdown of sugar. This is breakdown of sugar. You have prepared the recipe in the kitchen. Now it is time for you to utilize, and mitochondria will convert it into energy in the form of ATP. So mitochondria, what is the function of mitochondria? Aerobic respiration. Can you tell me the process under this two? Which organelle shows catabolic process and which organelle shows anabolic process? Type it in the comment section. Okay, correct answer, or even whatever the answer you know. Respiration, anaerobic respiration. Function of chloroplast, photosynthesis. Function of chloroplast is photosynthesis. Now we'll quickly go through the whole, and we'll write it once. That is, first is the vacuole, the tonoplast. Single membrane of vacuole is called as tonoplast. Plant vacuole occupies 90% of the total cell volume. Food vacuole is seen in Protista, and contractile vacuole is seen in Amoeba, which helps in osmoregulation and excretion of Amoeba. Then semi-autonomous organelles or mitochondria and chloroplast, they are called as semi-autonomous organelles because they have their own circular DNA, own 70S ribosomes. What are the four similarities between the two organelles? Double membrane, circular DNA, 70S ribosomes, and these ribosomes are also seen in prokaryotic organisms. That is the reason they are included in endosymbiotic origin. Okay, these two organelles are included in endosymbiotic origin. Next you have this one, that is eukaryotic cells contain cytoplasmic 80S ribosomes, that is ribosome organelles, mitochondria 70S, chloroplast 70S. Next, the primary function of mitochondria is aerobic respiration and chloroplast is photosynthesis. Meaning of aerobic respiration is sugar is converted into ATP, that is ATP synthesis. Next, photosynthesis is sugar synthesis. This one is the ATP synthesis, is the breakdown of sugar. This aerobic respiration is the breakdown of sugar. Okay, aerobic respiration happens in the presence of oxygen. This is also an important point. So shall we write it down without seeing the cheat sheets? Okay, students, get your paper. We'll start. So today the first one, what we saw in block 1, vacuole. The single layer membrane surrounding the vacuole is called as tonoplast. Next, vacuole occupies the 90% of total cell volume. Then food vacuole is seen in Protista. Are you able to write it down? Students, are you able to write it down? Are you able to remember by this method? You have to do it with focus. Contractile vacuole is equal to, that is, it is seen in amoeba. Next, contractile vacuole helps in osmoregulation and excretion. Are you able to finish it off till this point, or do you forget any particular point? Next, you have block 2. Block 2. What are the organelles? Semi-autonomous organelles. Semi-autonomous organelles are, what are those two organelles? Mitochondria and chloroplast. Okay. Next, what are the similarities both organelles have? Quickly, can you say? First we'll write down the reason for semi-autonomous. Okay, 70S type ribosomes, and they have circular DNA. Then what is the third point we saw? Okay, double membrane. Then both have relationship to endosymbiotic origin. What is the function of mitochondria? We saw ATP synthesis, that is aerobic respiration, conversion of sugar to ATP. It is a breakdown process. Breakdown of sugar. I asked you which one the type of metabolism in both. Okay, which one shows anabolism and which one shows catabolism? Quickly type the answer. Then chloroplast. Function of chloroplast we saw. That is photosynthesis. Photosynthesis. Which point I left out? Are you able to tell me which point I left on point? Okay, just now I am able to remember. Chlorophyll photosynthesis, it is sugar synthesis. This sugar is converted here into ATP and used as energy. This is aerobic respiration, which happens in the presence of oxygen. Okay, so which point I left out? Okay, which point we left out? We left out the very important point, that is the NEET trap. Eukaryotic cells contain what? How many types? The cellular ribosome is 80S type. The semi-autonomous organelles, they are what? Mitochondria and chloroplast. They contain 70S type of organelles, which means an eukaryotic cell 80S to 70S types are present. This can be asked as an AR type of question. Okay, these examples can be asked as match the following or a direct question. TonoplaST are on direct question or match the following. Okay, so then what you learned today, one mental hack is to break anything, to break your fear, to build confidence, or to master anything. Repetition is the key, one of the key to success. Okay, I've heard a sentence by the great martial artist Bruce Lee, that is, I don't fear a man who knows 100 kicks, but I fear a man who practiced the same kick 100 times. The same kick 100 times, which means he has mastered a single kick. Okay, a single kick can blow a person, right? So that is where is the secret. Okay, students, I really enjoyed the session. Even I learned myself everything back again. And tomorrow we will see with interesting code words for NEET again. Stay tuned with NEET. Thank you for listening and patiently writing with me. Have great days ahead.